Hello and welcome to this Microbiology Bio 203 video lecture on Chapter 3 from your OpenSTAX book, The Cell. I'm Mr. Kennedy and I'll be your guide as we explore this topic together. As we begin our journey through the cell, we will take a brief dovetail to examine the great debate between spontaneous generation and biogenesis, which started hundreds of years ago with a man by the name of Aristotle, who was alive between 384 and 322 BC. In Aristotle's time, many people believed that life could arise from non-living matter. Aristotle himself proposed that pneuma, or vital heat, was responsible for imparting life into non-living things. This feeling or idea was based largely on the fact that society as a whole during Aristotle's time lacked the basic scientific knowledge that most of us today possess. They had no way of explaining simple facts or events like maggots appearing out of decaying meat or mice showing up in a basket of grain. With their limited scientific background, it was their feeling that the mouse may have somehow hatched from the grain itself. Life came from a non-living thing. Oddly enough, this idea persisted well into the 17th century. During the 17th century, many scientists finally began to question this theory. One of the first scientists to do so is a man by the name of Francisco Redi, who in 1668 performed a classic experiment using meat and jars. In Francisco Redi's experiment, as seen below, he placed chunks of meat into different types of jars. Some were left open, others were sealed with cork, and others had a lid made of gauze. Over the course of the next several days, the meat began to spoil and attract flies. In the jars where the flies could come in direct contact with the meat, maggots appeared shortly thereafter. In the corked and covered jars, the meat never developed any maggots. The only other incidence of maggots appeared on the jars that were covered with gauze, and those maggots were only on the gauze. This refuted the idea of spontaneous generation, or the idea in this case that maggots were somehow born out of decaying meat, and proved that maggots, were the offspring of flies, and only when flies could come in direct contact with the meat did we see any maggots at all. There were other experiments during this time period that may appear to have supported spontaneous generation. One in particular was performed by John Needham. Needham put beef broth into glass flasks, heated it for a short time, and put a cork in the flask. Over the course of the next several days, Needham's broth always spoiled, in his mind supporting spontaneous generation. There were a couple of flaws in Needham's experiment, in that he did not boil the broth long enough to sterilize it, nor did he take into account the fact that a cork is porous and air could enter into his broth. A second scientist refuted Needham's work. His name was Lazzaro Spallanzani. Spallanzani repeated Needham's experiment with a slight change in that he heated the broth longer and sealed his flask with melted glass. His broth never spoiled. The primary issue with Spallanzani's work is it proved that as long as air doesn't touch your broth, then life won't appear in it. This led people to believe that there was a vital force still floating in the air that could impart life to non-living things. Enter Louis Pasteur. Louis Pasteur is credited for ending the great debate between competing groups who believed in spontaneous generation or those who believed in biogenesis. In Louis Pasteur's case, he took a flask and filled it with beef broth, but this was no ordinary flask. It was a flask with what is referred to here as a swan neck, or S-curved neck. This long, curved neck allowed him to put broth into the flask and boil it it also allowed air to enter and exit the flask. As he boiled the broth for more than 20 minutes, he sterilized it. He then left it sitting on his lab bench, and over the course of weeks, the broth never spoiled. Air could move into and out of the flask, but it had to do so ever so slowly. As it moved slowly, it could no longer suspend or carry anything in it. As the air passed the first curvature in the swan neck, the material that it was carrying settled out. By the time the air reached the broth, it was in effect sterilized, and subsequently his broth remained sterile as well. To prove that there was no vital force and that it was in effect the material that had collected in this curve of his swan-necked flask that was causing spoilage, Louis Pasteur took his flask and tipped it on its side so that the broth could come in contact with this material. He took another flask and actually broke the neck off, and within a few hours, the broth began to spoil. This disproved spontaneous generation once and for all. We now follow the idea that living things must come from other living things, or biogenesis. At this point, we'll look at what is referred to in your text as the cell theory and the men who are responsible for its inception. We begin by looking at Robert Hooke. Robert Hooke is credited with being the person to derive the name cell for cells. He observed slices of cork under a microscope. His simple microscope had about a 10 times magnification, and the image that he was able to see is on the right side of this slide. He described what he saw as honeycombed structure, or little rooms that again he named cellulae, and we today refer to as cells. The cells in cork are dead, so no other internal structure of the cell was visible to Robert Hooke. Furthermore, Robert Hooke never saw living material under his microscope. That credit, you might remember from a previous video, went to Anton von Leeuwenhoek. Further developing the cell theory, Schwann, Schleiden, Remek, and Virchow contributed a great deal of information. Schwann and Schleiden separately described animal and plant cells in the 1800s. As they studied these tissues, they realized there were similarities between them, and that cells were the fundamental components of both tissue types. Robert Remek is responsible for deriving the idea that cells must arise from other cells via cell division. Virchow later supported this idea. One problem with this premise is that if all cells arise from other cells via cell division, then where did the first cell come from? Even with the modern technology of today, we are still unable to answer that question. As microscopes improved and study continued, organelles were found in cells. Robert Brown is credited for describing the nucleus in a plant cell, while Andreas Schrimpner is credited with describing or discovering the chloroplast. As organelles were discovered in cells, many people began to ask, where did they come from? This led to what we now refer to as the endosymbiotic theory. The endosymbiotic theory is meant to explain the appearance of mitochondria and chloroplasts in cells of today. Mitochondria and chloroplasts are unique organelles in that they have their own DNA and their own ribosomes, and they can divide independently of the rest of the cell. This leads people to believe that at one point in a mitochondria or chloroplast's life history, it may have lived independently. If we follow this idea and theory, this means that, again, at some point in cells' timeline, that cells lacked these structures. And as populations grew, food became scarce, cells started to eat other cells. One of these cannibalistic cells, if you will, potentially swallowed a mitochondria and somehow realized that the mitochondria was producing energy. So rather than digesting the mitochondria as it would a normal food item, it simply held the mitochondria trapped in its body and passed it on to all future generations of itself. Exactly how it made that decision, no one knows. That's the theory of endosymbiosis. That brings us to germ theory of disease. As we learned more about microorganisms, we began to question if they had a role in making us sick. From the ancient Greeks, who proposed miasma was responsible for disease, through Semmelweis, who observed a connection between pleural fever and no hand washing, to eventually Robert Koch, we have learned a great deal about the connections between microorganisms and the diseases they cause. You might remember Koch's postulates, which were mentioned in a previous video, which are still used today to connect specific bacteria to the diseases that they cause. This is a timeline of the contributions of the scientists that I've mentioned previously. Next, we're going to examine the world of prokaryotes and eukaryotes and work our way slowly through the structures found in each. Prokaryotic cells do not contain internal membrane-bound organelles. They have plasma membranes and ribosomes. They have a region called the nucleoid where DNA can be found. This is not a nucleus because the DNA is not bound by a membrane. Almost all prokaryotic cells have a cell wall and are about 10 times smaller and grow faster than their eukaryotic kin. This slide will provide you with a brief comparison of a prokaryote to a eukaryote. Again, prokaryotes have one circular chromosome or DNA in a nucleoid, whereas a eukaryote has a paired chromosome in a nucleus, which is membrane-bound. Prokaryotes have no histones, eukaryotes do. Prokaryotes have no organelles that are membrane-bound, and eukaryotes do. Prokaryote cell walls often have peptidoglycan or pseudomirin, while eukaryotic cell walls have polysaccharides. Prokaryotes will divide by binary fission, and eukaryotes will divide by mitosis or budding. Prokaryotic cells may also take on a variety of shapes. This slide illustrates a variety of those shapes, including rod-shaped, which is bacillus, round, which is coccus, or a twisty shape, which is referred to here as spirillum. Prokaryotic cells can also pair or form chains or clusters, and this too adds to their name. When paired, we use the prefix diplo to describe the arrangement. In chains, we use the prefix strep. In groups of 4, we refer to them as tetrad. In groups of 8, we refer to them as sarcinae. And when in grape-like clusters, we use the prefix staph. We can connect multiple terms to describe the full arrangement of cells in this way. Looking at bacillus, who typically are single rods, should they pair up, we use the term diplobacilli. Should they form chains, we can use the term streptobacilli. Understanding the connections in terminology to shape can help you predict what you should see under a microscope when you look at them. The structures of the bacterial cell. The structures of the bacterial cell outside the cell wall include things such as the fimbriae, pilus, capsule, and flagellum. Other structures of the bacterial cell, again, include the cell wall, the cytoplasm, and the plasma membrane. As we work through the prokaryotic cell, we begin with the nucleoid. The nucleoid region is not bound by a membrane. The DNA is organized in one or more chromosomes and is circular. There are naps in the nucleoid, which is a nucleoid-associated protein. Archaea have naps or histone-like DNA, which organize their proteins. Plasmids. Plasmids are small, extra-chromosomal circular pieces of DNA. These circular pieces of DNA replicate independently of the chromosome, and they usually contain anywhere from 5 to 1,000 genes. they can be passed from one cell to the next in a form of bacterial sexual reproduction called conjugation. They can even be harvested by research scientists and used in transformation experiments. Ribosomes. Ribosomes are the sites of protein synthesis. They're composed of two subunits, each composed of protein and rRNA. Prokaryotic ribosomes are smaller and less dense than their eukaryotic cousins. Many antibiotics work by inhibiting protein synthesis on prokaryotic ribosomes. There are a variety of inclusions also found inside prokaryotic cells. Inclusions might include cytoplasmic structures to store nutrients, or inclusions that do not store nutrients but instead contribute to buoyancy or carbon fixation. A variety of inclusions are listed for you here. Endospores. Endospores are structures that are formed by some gram-positive bacteria under nutrient-stressed conditions. I prefer to refer to an endospore as almost a microbial lifeboat. They are packets of genetic material that are highly durable and resistant to dehydration and a wide variety of chemicals. Should situations become stressed for the bacteria and the microorganism die, the spore or endospore is likely to survive. From that endospore, a new generation of bacteria can grow once conditions prove favorable. Sporulation is the process of forming an endospore. It generally occurs when a key nutrient such as carbon or nitrogen becomes scarce or unavailable. A spore is formed inside the bacterial cell. It can be in the middle or to one end, and it is simply DNA packaged in a tough, protective outer coating. When done, the vegetative cell will disintegrate and the endospore is free. Germination. A spore can return to being a vegetative or growing cell by a process called germination. It is triggered by physical or chemical damage to the spore coat and forms a single cell. Endospore formation is not a form of reproduction. It is a survival system. Next, we have the plasma membrane. The plasma membrane is made up of a phospholipid bilayer. It also contains peripheral, integral, and transmembranal proteins and regulates what goes into and out of a cell. Cell membrane transport mechanisms include simple diffusion, osmosis, facilitated diffusion, active transport, group translocation, and engulfment. In simple diffusion, particles will move from an area of high concentration to an area of low concentration. In osmosis, we get the diffusion of water. In facilitated diffusion, diffusion occurs but utilizes carrier proteins as the particles that are moving are often too large or charged to pass through the membrane on their own. In active transport, energy and carrier proteins are required, and we often go against the concentration gradient, moving things that are large and charged. Group translocation occurs only in bacteria, mainly for sugars, and engulfment occurs only in eukaryotes. Photosynthetic membrane structures. Some bacteria can photosynthesize. Examples include cyanobacteria. Cyanobacteria and other photosynthetic bacteria have infoldings of the membrane that contain pigments called chromatophores, and these chromatophores function in a way as to absorb the sun's energy and perform photosynthesis as we understand it in a eukaryotic chloroplast. The cell wall. The cell wall is a semi-rigid structure. It adds shape, protects the cell from rupture when osmotic pressures are too high. It can be an anchor point for flagellum, and clinically important because it contributes to the ability of some organisms to cause disease. Chemical composition of the cell wall in a prokaryote often involves peptidoglycan and or pseudomurin. In cases where peptidoglycan is utilized, either alone or in combination with other substances, the cell wall structure is seen in repeating units of the disaccharide N-acetylglucosamine and N-acetylmuramic acid, illustrated below. Gram-positive cell wall structure. A gram-positive cell has a very thick cell wall, made mostly of peptidoglycan, held together and stabilized by teichoic acid, whereas a gram-negative cell has a thin cell wall sandwiched between two plasma membranes, seen in these illustrations. A little more information on the gram-negative cell wall is here. It has a thin peptidoglycan with no teichoic acid, a paraplasmic space, an outer membrane made of lipopolysaccharides. And that outer membrane provides protection from phagocytes, complement proteins, and antibiotics. Here is a simple comparison between a gram-positive cell wall and a gram-negative cell wall. Again, the gram-positive has thick peptidoglycan, teichoic acids, and is more resistant to mechanical forces. The gram-negative cell wall has thin peptidoglycan in the paraplasma between the outer membrane and plasma membrane. It is more susceptible to mechanical breakage, more resistant to antibiotics and the immune system, and the outer membrane can provide protection and is a pathogenic factor. Gram staining takes advantage of the differences between the two cell walls. As you'll learn in lab, gram staining utilizes a combination of crystal violet, Gram's iodine, a decolorization or destaining alcohol, and a counterstain known as safranin. The primary stain, crystal violet, stains both gram-positive and gram-negative cells purple. The mordant, Gram's iodine, causes larger amounts of crystal violet to adhere to these cells. The destaining process of alcohol washes away excess crystal violet. Gram-positive cells with the thick peptidoglycan cell wall are not fully decolorized. The crystal violet remains. In gram-negative cells, the alcohol dissolves the outer membrane, which was infused with crystal violet, and the crystal violet is washed away. The counterstain, safranin, stains the cells pink. A gram-positive cell, which has already absorbed copious amounts of crystal violet, will still stay purple, whereas the gram-negative cell, whose crystal violet was washed away, will become pink. There are a few atypical cell walls in the world of bacteria. One case is the acid-fast bacteria. Basically, acid-fast bacteria are gram-positive. They have a waxy lipid or mycolic acid bound to their peptidoglycan. Some examples include Mycobacterium and Nocardia. This waxy pellicle makes it hard to penetrate the cells. They are very resistant to antibiotics, as is the case of things like tuberculosis. These cells are also very slow growing. Mycoplasm and archaea also have atypical cell walls. Mycoplasm have no cell wall, instead infuse their plasma membrane with sterols, and the archaea could be wall-less, or they may have a cell wall of pseudomurin, which lacks the NAM and D amino acids. Glycocalyces and S layers. The glycocalyx is a sugar coat. It's sticky, and it comes in two types. When organized, we refer to the glycocalyx as a capsule. When unorganized and loose, we refer to it as a slime layer. The glycocalyx helps with adhesion and biofilm formation. Capsules protect from desiccation and phagocytosis. The S layer is made up of proteins and glycoproteins, which provide protection and interaction with the immune system. Filamentous appendages, such as fimbriae and pili, are hair-like structures that are made of protein called pilin and arranged helically around a central core. Fimbriae allow attachment to surfaces, while pili, usually only 1 to 2 per cell and are longer than fimbriae, can join two cells to allow DNA movement from one cell to another through bacterial conjugation. The flagellum is a long filamentous appendage used for movement and is illustrated here. Flagellar arrangements can be found in different types of cells and varies from cells who lack flagellum altogether to cells that have flagellum covering their entire surface. Axial filaments are also called endoflagellum and are found in spirochetes. These are anchored at one end of a cell, and the rotation causes the cell to move. Next, we have eukaryotic organisms. Eukaryotic organisms have flagella that are longer and more complex. They can have cilia, which are shorter than flagellum. They have cell walls, typically in plants, made with carbohydrates. They have a cytoplasmic membrane, which is similar to prokaryotes and also contains sterols. They have a cytoskeleton, which is an internal structure facilitating movement, and a variety of organelles. This is a picture of a generic eukaryotic cell. Be sure to familiarize yourself with each labeled structure. Cell morphology can vary in eukaryotic cells as it did in prokaryotic cells. Eukaryotic cells may be spheroid, ovoid, cuboidal, cylindrical, flat, lenticular, fusiform, and so on. Eukaryotic cells have a nucleus. The nucleus is a structure possessing DNA or genetic material enclosed in a nuclear envelope. It typically is the largest organelle in a eukaryotic cell. Inside the nucleus is a nucleolus. This is a dense region responsible for rRNA synthesis. rRNA ultimately condenses to form ribosomes. The ribosome. Ribosomes in eukaryotes are larger than their prokaryotic kin. They differ from prokaryotic ribosomes not only in their size, but also in the fact that they may be free-floating or attached to the endoplasmic reticulum. In either case, the function is still the same. Ribosomes make protein. Eukaryotic cells have an endomembrane system. This is a series of membranes, or sacs, and the site of biosynthesis in the cell. The endomembrane system includes the endoplasmic reticulum, which comes in both smooth and rough varieties. The smooth endoplasmic reticulum produces phospholipids, while the rough produces protein. The Golgi apparatus is used for packaging and distribution in the cell. Lysosomes and perioxysomes. Lysosomes contain digestive enzymes which break down particles, microorganisms, and damaged organelles. Perioxysomes are not part of the endomembrane system, and they function in lipid biosynthesis and the breakdown of other molecules. The cytoskeleton. This is a scaffold of the cell, and it is involved in cell shape, motility of the cell. The cytoskeleton includes microfilaments, intermediate filaments, and microtubules, as illustrated here. The mitochondria and chloroplasts. Mitochondria and chloroplasts both harvest energy and make ATP. The mitochondria harvest their energy from food and release it to the rest of the cell in the form of ATP through cellular respiration. Chloroplasts harvest energy from the sun through photosynthesis. Both are believed to have been bacteria before becoming endosymbionts to eukaryotic cells, again because they have their own DNA and ribosomes and can divide independently of the rest of the cell. This is an illustration of a basic chloroplast. The plasma membrane of a eukaryote, like the plasma membrane of a prokaryote, is a phospholipid bilayer and also contains proteins, integral and peripheral. It contains cholesterol to add to its stability, and as its prokaryotic kin, controls what goes into and out of the cell. Membrane transport mechanisms for a eukaryotic cell are again similar to what we saw in a prokaryote. Simple, facilitated diffusion, osmosis, active transport are all the same. Unique to eukaryotes include endo and exocytosis, the use of invagination and vesicle formation to take in large objects and digest them. This is an illustration of cilia and flagella. They are used for locomotion or to move substances along the cell surface. Flagella differ from those of prokaryotes in that they are flexible and whip-like and made of microtubulin. Cilia are unique to eukaryotes and are shorter versions of flagellum. The cell wall and extracellular matrix is last. Fungi, plants, algae, and some protists have cell walls. This is different from a prokaryote. The extracellular matrix is composed of protein and a carbohydrate component. This protects cells from physical stress and transmits signals arriving at the outside edges of the tissue to cells deeper within the tissue. And this concludes our coverage of Chapter 3, the cell.